Organization of CVS, Part 1. Introduction to the CVS. Cardiovascular system transport essential nutrients, oxygen, hormones to the tissues and remove the product metabolism from the tissues. Also contribute to the regulation of many homeostatic functions like fluid balance, temperature regulation of the body. Cardiovascular functions are carried out primarily by altering the amount of blood pumped by the heart. Component of the CVS, central pump, the heart, and the circulatory system, the blood vessels. So here's the first component of the CVS, central pump, the heart. Primary function of the heart is to pump blood into the circulation. Consists of a dual pump, heart made up of a dual pump, right pump and left pump. Right ventricle that propels blood into the pulmonary circulation for exchange of gases in the lungs. Left pump, left ventricle that propels blood into the systemic circulation to supply oxygen and nutrients to the tissues. Heart has four chambers, two atria and two ventricles. Here you can see here the right side of the heart and here is the left side of the heart. And this is the right atria, this is the left right ventricle. And here is the left atria and here is the left ventricle. Right atrium pumps the blood into the pulmonary circulation. Right ventricle pumps the blood into the pulmonary circulation. Left ventricle pumps the blood into the systemic circulation. Circulatory system, the blood vessels. Blood is distributed to the different part of the body by systemic arteries. And the pulmonary artery is used for the pulmonary circulation, where the blood get oxygenated through lungs. Systemic arteries are more extensively branched and thicker than the pulmonary artery. So therefore, the systemic circulation provides more resistance to flow of blood. Left ventricular pumps blood at much higher pressure than the right ventricle. Because of this, the left ventricle get hypertrophied, the muscle mass got increased, and the shape of the left ventricle is the circular shape. And right ventricular pump blood at a lower volume, lower pressure. That's why its shape is semilunar and the muscle not get hypertrophied. Peak left ventricular pressure is 120 mmHg and the peak right ventricular pressure is 25 mmHg. Blood vessels. Blood vessels are three layers. Blood vessels are made up of three layers mainly. One is the tunica externa, that it contains elastic tissue and collagen fibers. Collagen fiber and the tunica media. Second layer is the tunica media. Consists of smooth muscles and elastic tissues. Smooth muscles and elastic tissues. Tunica media, smooth muscles are arranged circularly and the lumen of the blood vessels, around the lumen of the blood vessels. So when sympathetic stimulation, the circular muscle got contracted, and because of this, vasoconstriction occur. And when sympathetic inhibition occur, muscles get relaxed and vasodilation occur. And the outer innermost layer is the tunica intima, that is composed of endothelial cell lining, which is a simple squamous epithelium. Tunica intima just made of endothelial cell lining that helps in exchange of product between blood vessels and the tissues. And the component of blood vessels. There are mainly the four components from form the vascular wall. The endothelial cells. Endothelial cells, all blood vessels are made up of endothelial cells, mainly endothelial anterior single lining that help in the exchange of component between blood vessels and the tissues. Next is the elastic tissues. Elastic, the more the elastic tissue present in the vessel wall, more it will be the more it has the compliance and stretchability property. The large arteries and aorta mainly are more compliant as they have more elastic tissue component in them than the others. Smooth muscles, smooth muscles present in arterioles, meta-arterioles. They are more in number than the elastic tissue. That's why arterioles, meta-arterioles are not more compliant. And the fibrous tissue, mainly the collagen fibers. They also provide tension and reduce the elastic property of the vessels. Distribution of these components in various blood vessels determine their functional significance, as I described. And here you can see in the diagram, aorta, they have endothelial cells. All have endothelial cells in them, but elastic fiber are absent in capillary and Elastic fiber absent in capillary and venules. And smooth muscles are also absent in capillary and venules. Elastic fiber is more in aorta than the smooth muscle in a fibrous tissue. But in medium arterial, smooth muscle component is more. That's why arterial and medium artery provides maximum resistance to blood flow. And the fibrous tissue, here you can see fibrous tissue component.
CVS Part 2 by Karthik Thakur. So we have physiological classification of blood vessels. First of all, we have Windkessel vessels. Windkessel is a German word which here implies elastic recoil. The aorta and large arteries here are called as Windkessel vessels. Since the aorta and large arteries have more elastic elements in their wall, therefore these vessels have higher compliance. Having higher compliance means their ability to expand without recoiling back to their original position. The Windkessel or the recoiling effect pushes blood in forward direction during diastole. As in systole, the blood pressure increases, it makes the arteries to expand more, and during diastole, they just pushes the blood in forward direction. Now, second type of vessels, we have resistance vessels. Arterioles, meta-arterioles, and smaller arteries are the resistance vessels. Since as the arteries go towards periphery, they divide into arterioles, meta-arterioles, and smaller arteries. These provide the resistance and are known as the resistance vessels. They have thick muscle coat and less elastic fiber in their wall. Therefore, they provide resistance to blood flow. Now, we have exchange vessels. Capillaries are here known as exchange vessels because their wall is formed only by a single layer of endothelial cells, due to which the blood flow is sluggish in capillaries and, and due to more total cross-sectional area of these vessels. These two factors, which is thin wall and sluggish flow, they favor exchange along the capillary walls and hence they are known as exchange vessels. Now we have capacitance vessels. Capacitance vessels here refers to a reservoir. Normally, more than 60% of the total blood is present in the venous compartment, and veins can accommodate still larger amount of blood without increasing much pressure in them. Therefore, large and small veins are called as capacitance vessels due to their capacity of holding blood. Now we have divisions of vascular system. The vascular tree here consists of three systems: arterial system, capillary networks, and venous system. Now coming toward arterial system, we have arteries, arterioles, and meta-arterioles. Arteries are the biggest among these, and they have a thick tunica media, which is the middle layer of an blood vessel. The smooth muscle of arteries are extensively innervated by sympathetic fibers of ANS. Now we come to arterioles. As arteries move forward to the periphery of our body, they divide into arterioles and meta-arterioles. Now we have arterioles. Arteries branch out to form the arterioles. Arterioles regulate the resistance to flow and control the distribution and rate of blood flow to the organs. They have thick muscle coat and few elastic fibers in their wall. The ratio of thickness of the wall to the diameter of vessel is very high in arterioles. Now coming to meta-arterioles, the arterioles terminate in meta-arterioles, and a flap of smooth muscle called precapillary sphincter is present at the end of meta-arterioles or arterioles. Now coming to capillary networks, the system of capillary network originates from arterioles and meta-arterioles. Precapillary sphincter, which is present in meta-arterioles and arterioles, control the amount of blood flow into the capillaries. Now coming to venous system, we have venules and veins present in it. Venous system starts from capillaries. Venules, many capillaries unite to form venules. The function of venules is to collect blood from capillary and drain it into the veins. Now veins, we have venules uniting to form veins and that again join together to form larger veins. Veins serve as the blood reservoir, and yeah, we have discussed that it is also called as capacious vessels. The venous valves are made up of thin folds of tunica intima that forms a flap-like cusp. These valves point toward the heart and prevent retrograde flow of blood. Thus, venous valves facilitate venous return. So, to summarize it, we have arteries that, as they move toward any organ or periphery, divide into capillary, and these capillary then unite to form venous system. Now, to give an overview between systemic and pulmonary circulation, we have systemic circulation, which is also called as greater circulation or peripheral circulation, since greater part of our body is supplied by systemic circulation. And pulmonary circulation is called central or lesser circulation, since only lungs are supplied by pulmonary circulation. Pressure in systemic circulation is very high, while in pulmonary circulation this is low pressure. The arteries of systemic circulation and arterioles have thick wall and narrow lumen, while in pulmonary circulation they have thin wall and large lumen. The capillaries of systemic circulation are long and narrow, while those of pulmonary circulation are short and wide. Resting sympathetic tone. High vasoconstrictor tone is present in systemic circulation, while it is absent in pulmonary circulation. The peripheral resistance in systemic circulation is high due to presence of more arterioles and meta-arterioles, while it is low in pulmonary circulation. Now, the volume distribution in vascular compartments, it is 60% in systemic veins and venules. Then we have systemic arteries, which contain 12 to 15%. Then pulmonary vessels, which contain 10 to 12%. Heart, which contain 5 to 10%. And systemic capillaries, which contain 5%. Thank you.
So the topic for today's test seminar is organization of the CVS. The question covered in this seminar is are differentiate between the systemic and pulmonary circulation and explain the basic principle of circulatory function. Today's seminar is presented by me, Arun Mehra, roll number 18, and my fellow batchmate Ripu Daman, roll number 85. So first of all, what is the meaning of systemic circulation? The circulation of blood through a systemic circular bed is called systemic circulation. This is also called peripheral circulation. This is a major circulation as it contains 80 to 85% of total blood volume. It provide oxygenated blood to different parts of the body and drain venous blood back to heart. The left ventricle pump blood into the systemic circulation. The pressure in the systemic circulation, especially in arterial compartment, is high. The systolic pressure is 100 to 140 mm of Hg. The pressure drop progressively as the blood flows from arteries to vein. The arteries and arterioles have thick walls and narrow lumen and highly richly innervated by sympathetic fibers. They have high resting vasoconstrictor tone, thus they provide higher resistance to blood flow. The capillaries are longer and narrower, therefore systemic circulation also provides resistance to blood flow.
Now we will see the pulmonary circulation. So the question is that what is pulmonary circulation? The circulation of blood through the pulmonary vascular bed is called as pulmonary circulation. This is also called the central circulation because it is the minor circulation as it contains only 10 to 12% of the total blood volume. It provides a low resistance pathway for the entire output of the right ventricle to traverse through the lungs. The primary function of pulmonary circulation is to exchange gases between the blood and the atmosphere. The arteries have thin wall and large lumen. Therefore, pulmonary arterioles provide low resistance to flow. They have also rich sympathetic innervations, but unlike that of systemic arteries, they lack resting vasoconstrictor tone. The capillaries are shorter and wider. Consequently, resistance is also less in capillaries. The vessels in the pulmonary circulation are primarily designed to provide a low resistance circulation for the entire output of the right ventricle to pass through them. The output of the right ventricle is same as the output of the left ventricle. The blood from the left ventricle is pumped into the systemic circulation, which is a much larger compartment that easily accommodates the left ventricular output. The equal amount of blood is ejected by the right ventricle but into a much smaller compartment, the pulmonary circulation. Now, we will see the basic difference between the systemic and pulmonary circulation. The blood flow, which is seen in liter per minute, is 5 liter per minute in systemic circulation and similarly 5 liter per minute in pulmonary circulation. Percentage of blood volume is 84 in case of systemic circulation and 9 in case of pulmonary circulation. Arterial blood pressure in mm of Hg is 120 by 80 in case of systemic circulation and 25 by 8 in case of pulmonary circulation. The resistance is high in systemic circulation and low resistance is offered to the blood in case of pulmonary circulation. Oxygen content of arteries, that is the partial pressure of oxygen in mmHg, is 100 in systemic and 40 in pulmonary circulation. There is vasodilatory effect in systemic circulation in case of hypoxia and vasoconstriction in case of pulmonary circulation.
So now the basic principle of circulatory function. Although the detail of circulatory function are complex, the basic principle underlie all these the function. Blood flow to most tissue is controlled according to the tissue need. When tissue are active, they need increased supply of nutrient. Therefore, more blood flow than when at rest. Occasionally, at much as 20 to 30 times the resting level. However, the heart normally cannot increase its cardiac output more than 4 to 7 times higher than the resting levels. Therefore, it is not possible simply to increase the blood flow everywhere in the body. When a particular tissue demands increased flow, instead of microvessels of each tissue, especially the arterioles, continuously monitor tissue needs such as availability of oxygen and other nutrients and the accumulation of CO2 and other tissue waste products. These microvessels in turn dilate or constrict to control local blood flow at the level required for the tissue activity. Also, nervous control of the circulation from the central nervous system and the hormones provide additional help in controlling tissue blood flow. Cardiac output is a sum of all the local tissue flow. When blood flows through the tissue, it immediately returns by the way of veins to the heart. The heart responds automatically to the increased inflow of blood by pumping it immediately back into the arteries. Thus, as long as the heart is function normally, it acts as an automaton, responding to the demands of tissue. The heart, however, often needs help in forms of special nerve signals to make it pump the required amount of blood flow.
Now, the arterial pressure regulation is generally independent of either local blood flow control or the cardiac output control. The circulatory system is provided with an extensive system for controlling the arterial blood pressure. For example, if any time the pressure falls significantly below the normal level of about 100 mmHg, a barrage of nervous reflexes elicits a series of circulatory changes to raise the pressure back toward normal within the second. The nervous signal especially do the following: increase the force of heart pumping, control contraction of the large venous reservoirs to provide more blood to the heart, cause generalized constriction of the arterioles in many tissues so that the blood accumulates in the large arteries to increase the arterial pressure. Then over more prolonged periods, hours and days, the kidney play an additional major role in pressure control by secreting pressure controlling hormones and regulating blood volume.
Thank
Thank
you.
you.
Hello everyone. So today's topic is property of cardiac muscle part B, and the question covered in whole PPT is compare the structural and functional difference between cardiac, skeletal and smooth muscle. So basically what are muscle? Muscle is a soft tissue and it is one of the four basic tissues. Muscle are excitable tissue like the nervous tissue. Muscle help in movement, sport and protection of internal organs. Based on their location, three types of muscles are identified. First, skeletal muscle, smooth muscle and cardiac muscle. सबसे पहले हम देखते हैं मॉर्फोलॉजिकल प्रॉपर्टीज के बीच डिफरेंस इन ऑल थ्री मसल. तो पहला है डिस्ट्रीब्यूशन. स्केलेटल मसल अटैच बाय टेंडन टू द बोन्स. कार्डियक मसल ओनली हार्ट के अंदर प्रेजेंट होती है और ये बोन से अटैच नहीं होती है. स्मूथ मसल दो टाइप की होती है. पहला सिंगल यूनिट स्मूथ मसल कंसिस्ट ऑफ मल्टीपल सेल्स कनेक्टेड दैट कैन बिकम स्टिमुलेटेड इन ए सिंक्रोनाइज पैटर्न फ्रॉम ओनली वन सिनेप्टिक इनपुट. और ये प्रेजेंट होते हैं यूरेटर के अंदर, यूट्रस के अंदर, इंटेस्टाइन और ब्रोंकाई के अंदर. Second, a multi-unit smooth muscle differ from single unit in that each smooth muscle cell receive its synaptic input and this allow multi-unit smooth muscle to have much finer control. और ये प्रेजेंट होते हैं कैपिलरी मसल के अंदर, आइरिस इन आई, मसल इन ब्लड वेसल के अंदर. एंड सेकंड प्रॉपर्टी है स्ट्राइशंस. स्केलेटल मसल के अंदर वेल डेवलप्ड स्ट्राइशन प्रेजेंट होते हैं, कार्डियक मसल के अंदर भी प्रेजेंट होते हैं, लेकिन स्मूथ मसल के अंदर ये एब्सेंट होते हैं. तीसरा है साइज एंड शेप. स्केलेटल मसल का जो शेप है वो सिलेंड्रिकल होती है, कार्डियक मसल की भी सिलेंड्रिकल होती है, जबकि स्मूथ मसल की स्पिंडल शेप होती है. स्केलेटल मसल की बात करें तो इसके अंदर 1 to 40 mm लॉन्ग, 50 to 100 माइक्रोमीटर डायमीटर प्रेजेंट होता है और ये मल्टी न्यूक्लिएटेड होते हैं. जबकि कार्डियक मसल के अंदर 100 माइक्रोमीटर लॉन्ग होते हैं और 15 माइक्रोमीटर का डायमीटर प्रेजेंट होता है और ये ब्रांचिंग नेटवर्क बनाते हैं जिसके अंदर इंटरकलेक्टेड डिस्क एंड गैप जंक्शन भी प्रेजेंट होते हैं. जबकि स्मूथ मसल जो होता है वो सिंगल न्यूक्लियस वाला होता है और ये वेरिएबल साइजेस होते हैं. जैसा कि आप देख सकते हैं कि स्मूथ मसल जो होते हैं वो स्पिंडल शेप के होते हैं और उसके अंदर सिंगल न्यूक्लियस प्रेजेंट होता है, जबकि कार्डियक मसल जो होते हैं वो सिलेंड्रिकल शेप के हैं और इनके अंदर ब्रांचिंग नेटवर्क बना रहे हैं. एंड एज स्केलेटल मसल के अंदर भी स्ट्राइशंस प्रेजेंट है और ये सिलेंड्रिकल शेप के हैं और ये मल्टी न्यूक्लिएटेड भी है. एंड द फोर्थ प्रॉपर्टी इज सार्कोप्लाज्मिक रेटिकुलम, जो कि स्केलेटल मसल के अंदर वेल डेवलप्ड होती है और कार्डियक मसल के अंदर भी वेल डेवलप्ड होती है मोर देन स्केलेटल मसल, जबकि स्मूथ मसल के अंदर ये पूर्ली डेवलप्ड होती है. सो द नेक्स्ट इज सिंशियल स्ट्रक्चर. तो सिंशियम होता क्या कि अगर एक मसल को एक सेल को स्टिमुलेट किया जाए और उसके आसपास की सेल भी उसी सिग्नल से स्टिमुलेट हो जाए ऑटोमेटिकली, तो उसे सिंशियम कहते हैं. सिंशियल स्ट्रक्चर स्केलेटल मसल के अंदर एब्सेंट होते हैं, जबकि कार्डियक और स्मूथ मसल के अंदर प्रेजेंट होते हैं. तो नेक्स्ट इज सार्कोट्यूबुलर सिस्टम. स्केलेटल मसल के अंदर ये प्रेजेंट होता है. टी सिस्टम जो होता है वो एआई जंक्शन में प्रेजेंट होता है और टर्मिनल सिस्टर्न प्रोमिनेंट होते हैं, जबकि कार्डियक मसल के अंदर ये पूर्ली डेवलप्ड टर्मिनल सिस्टम प्रेजेंट होते हैं और टी सिस्टम जो होता है वो प्रेमिनेंट होता है और जेड लाइन के आसपास होता है, जबकि स्मूथ मसल के अंदर ये इतना ज़्यादा प्रोमिनेंटली प्रेजेंट नहीं होता है. जैसा कि आप यहाँ पे देख सकते हैं कि टर्मिनल सिस्टम्स और टी ट्यूब्यूल्स वगैरह यहाँ पे प्रेजेंट है. सेवंथ प्रॉपर्टी है नर्व सप्लाई. स्केलेटल मसल बेसिकली स्मोटिक नर्व के द्वारा सप्लाई की जाती है एंड कई स्पेशल नर्व भी इसे सप्लाई करती है, जबकि कार्डियक मसल एंड स्मूथ मसल बोथ सप्लाई की जाती है ऑटोनोमिक नर्वस सिस्टम के द्वारा. अगर कंट्रोल एंड रिदमिसिटी, तो स्केलेटल मसल जो होती है वो वॉलंटरी कंट्रोल होती है और उनके अंदर रिदमिसिटी प्रेजेंट नहीं होती है, जबकि कार्डियक मसल जो होती है वो कॉन्ट्रैक्ट करती है रिदमिकली एंड स्पॉन्टेनियसली इन द एब्सेंस ऑफ एक्सटर्नल इनर्वेशन बाय पेस मेकर जैसे ऐसे नोड. एंड स्मूथ मसल जो होती है उसके अंदर दो प्रकार की रिदमिसिटी पाई जाती है, एक रेगुलर एंड दूसरी होती है इरेगुलर. या तो ये रेगुलरली रिदमिसिटी मेंटेन करती है या फिर इरेगुलरली रिदमिसिटी मेंटेन करती है. इसके अगला है ब्लड सप्लाई एंड ऑक्सीजन कंजम्पशन. स्केलेटल मसल के अंदर 840 ml पर मिनट की मॉडरेट ऑक्सीजन कंजम्पशन होता है, जबकि कार्डियक मसल के अंदर 250 ml पर मिनट का हाई ऑक्सीजन कंजम्पशन होता है एंड स्मूथ मसल के अंदर 350 ml पर मिनट का ऑक्सीजन कंजम्पशन होता है. नेक्स्ट जो प्रॉपर्टी वो है इलेक्ट्रिकल प्रॉपर्टीज. तो रेस्टिंग मेम्ब्रेन पोटेंशियल ऑफ स्केलेटल मसल जो होता है वो -90 मिलीवोल्ट होता है, कार्डियक मसल का भी -90 मिलीवोल्ट होता है, जबकि स्मूथ मसल के अंदर इसकी रेंज वेरी करती है -30 मिलीवोल्ट से -50 मिलीवोल्ट तक, जो कि फ्लक्चुएटिंग होता है. एक्शन पोटेंशियल. स्केलेटल मसल के अंदर रैपिड डिपोलराइजेशन होता है विद द स्पाइक ड्यूरेशन इज 2 to 4 मिलीसेकेंड एंड टोटल एम्पलीट्यूड जो होता है वो होता है 120 मिलीवोल्ट का, जबकि कार्डियक मसल के अंदर रैपिड डिपोलराइजेशन होता है 15 to 20 मिलीवोल्ट का एंड एक्शन पोटेंशियल ड्यूरेशन होती है वो होती है 120 मिलीसेकेंड की और एम्पलीट्यूड जो होता है वो होता है 100 मिलीवोल्ट का. एंड स्मूथ मसल के अंदर जो वेरिएबल एक्शन पोटेंशियल होता है एंड द टोटल ड्यूरेशन इज 50 मिलीसेकेंड एंड टोटल एम्पलीट्यूड अप टू 60 मिलीवोल्ट होता है एंड वेरिएबल स्पीड ऑफ द कंडक्शन होती है. सो नेक्स्ट है मैकेनिकल प्रॉपर्टीज. इसमें है मैकेनिकल इवेंट्स. स्केलेटल मसल के अंदर जो कॉन्ट्रैक्शन स्टार्ट 2 मिलीसेकेंड आफ्टर द स्टार्ट ऑफ डिपोलराइजेशन बिफोर रिपोलराइजेशन एंड कैन सबमिट द कॉन्ट्रैक्टाइल रिस्पॉन्स एंड टेटनाइजेशन अकर. तो टेटनाइजेशन बेसिकली होता क्या है कि अगर किसी मसल को स्टिमुलेट किया जाए रिपीटेडली एट ए वेरी हाई फ्रीक्वेंसी तो उसके अंदर एक सस्टेन्ड कॉन्ट्रैक्शन आता है जिसको ही टेटनाइजेशन कहते हैं. कार्डियक मसल जो होती है उसके अंदर मोर देन हाफ ऑफ मैकेनिकल कॉन्ट्रैक्शन इज ओवर ड्यूरिंग एब्सोल्यूट रिफ्रैक्टरी पीरियड एंड टेटनाइजेशन नॉट सी. In the smooth muscle start to contract approximately 1200 millisecond after the start of the spike Peak contraction is reached approx 500 milliseconds after the spike, and thickness is seen. 13 is duration of muscle twitch. Varies with in the skeletal muscle, it is vary with the type of muscle fiber. 7.5 millisecond in fast muscle and 100 millisecond in slow muscle. In cardiac muscle, 1.5 times the total duration of action potential, and in the smooth muscle, approximately 1000 millisecond. And the next one is excitation contraction coupling. In skeletal muscle, the rapid process time from initial depolarization to initiation of contraction is 10 milliseconds. In cardiac muscle, more rapid process, and in the smooth muscle, it is very slow process. And the 15 number is the all and none law. What is basically all and none law? The strength by which a nerve or muscle fiber respond to a stimulus is independent of the strength of the stimulus. If that stimulus exceeds the threshold potential, that muscle fiber will give a complete response. एंड स्केलेटल मसल के अंदर ये एप्लीकेबल होता है एंड ट्रू होता है फॉर सिंगल मसल फाइबर. कार्डियक मसल के अंदर भी एप्लीकेबल होता है फॉर द होल आर्टरिया और वेंट्रिकल, एंड इन द स्मूथ मसल ये एप्लीकेबल होता है फॉर ए ट्रू सिंगल मसल फाइबर. 16 नंबर है लेंथ टेंशन रिलेशनशिप. In skeletal muscle, maximum active tension is developed at the optimum length. In the cardiac muscle, it is same as skeletal muscle, and in the smooth muscle, show property of plasticity. 17 है phenomena of fatigue. Phenomena of fatigue in the skeletal muscle is occur because it have very low refractive period, but in cardiac muscle it is not seen because उसका refractive period काफ़ी लंबा होता है comparatively. And in smooth muscle, it is possible the fatigue condition but difficult to demonstrate. And the last property is the metabolic properties. इसमें देखते हैं कि skeletal muscle जो होती है उसके अंदर जो एनर्जी वो यूज़ करते हैं अंडर basal state 20% by fats, more than 60% by carbohydrate and 20% by proteins. But the cardiac muscle जो यूज़ करते हैं वो करते हैं 65% by fats, 25% by carbohydrate and 10% by ketone bodies and amino acids. And the smooth muscle जो होते हैं वो low यूज़ करते हैं, mainly provided by the utilization of carbohydrate and fats. And thank you very much.
Hello everyone, my name is Atul, roll number 19. Our today's topic is properties of cardiac muscle. The part we will be covering is properties of cardiac muscle that ensures its non-fatigability. So what is muscle fatigue? Muscle fatigue is a decrease in maximal power production in response to contractile activity. Muscle fatigue is a symptom that decreases muscle's ability to perform over time. We can feel it in our daily life after we perform some rigorous activity or exercise like lifting heavy weight. Cardiac muscle do not fatigue though. It is truly amazing that the heart continues to beat in a healthy individual with such regularity the entire lifespan of an individual without getting tired. Even the most well-trained skeletal muscle in the most elite of athletes cannot achieve this. What are the reasons why cardiac muscle do not fatigue? The first and most important reason is cardiac muscle has a long refractory period. Cardiac muscle has a rich blood supply. They undergo aerobic metabolism, is rich in mitochondria. Long refractory period of cardiac muscles. The action potential in cardiac muscle is prolonged and has a long refractory period. Refractory period of the muscle is the period during which muscles does not evoke a response no matter how strong is the stimulus. The refractory period is divided into absolute and relative refractory periods. The absolute refractory period of ventricular muscle is about 200 milliseconds, and the relative refractory period is about 50 milliseconds. An important difference exists in the duration of action potential of cardiac and skeletal muscles. Duration of action potential of cardiac muscle is 250 milliseconds, which is almost same as the duration of the mechanical response. We will see it in a diagrammatic way further ahead. In skeletal muscle, the duration of action potential is maximum about 5 milliseconds, which is much less than the duration of mechanical response. So, in skeletal muscle, mechanical responses can be mushed and muscle can be tetanized, as the duration of action potential is much shorter than the duration of the mechanical contraction. Though in ventricular muscle the duration of action potential is almost same as the duration of mechanical response, the refractory period extends to the most part of the mechanical response. Therefore, in cardiac tissues, always a new action potential generates a fresh mechanical response. Consequently, a fresh contraction cannot occur before the completion of the previous mechanical response. Thus, mechanical response of ventricular muscle cannot be merged, and therefore cardiac muscle cannot be tetanized. It is to be noted, while tetany allows the generation of greater force, fatigue also sets in much faster. Here we have a relationship between electrical and mechanical responses in cardiac muscle and skeletal muscle. In cardiac muscle, due to long refractory period, a greater part of the mechanical response falls in the absolute refractory period of the action potential. We can see it in the diagram A. There is an additional relative refractory period of about 50 milliseconds during which the muscle is more difficult to excite than the normal. Nevertheless, it can be excited by a very strong excitatory signal like premature contractions. Though the premature contractions do not cause wave summation as occurs in skeletal muscle. In skeletal muscle, the mechanical response starts almost after the absolute refractory period, as we can see in the diagram B. Therefore, skeletal muscle can be tetanized but not the cardiac muscle. Other reasons why cardiac muscles do not fatigue. Cardiac muscle has a rich blood supply. In context, cardiac muscle has a capillary density that is about 4 times greater than the skeletal muscle. Cardiac muscle undergoes aerobic metabolism. Thus, the accumulation of lactic acid, an important factor in the development of fatigue in anaerobically exercising muscles, plays no role in cardiac muscles. Cardiac muscle is rich in mitochondria. Mitochondria account for almost 1/3 of the cardiac muscle by weight. Thank you.
So questions to be covered are action potential in conducting and contractile tissues, phases of action potential, and efflux during each phase of action potential. Action potential in contractile tissues. So what are contractile tissues? Contractile tissues are Purkinje fibers, atrial muscles, and ventricular muscles. They show fast action potential. Why they show fast action potential? Because they have fast sodium channels. They open very fast, so rapid depolarization occurs in it. Resting membrane potential in these tissue are about -85 to -90 millivolt. Slope of phase 4 in this is flat as compared to action potential in conductive tissue. These tissue does not show the property of autorhythmicity because of overridden suppression of SC node and absence of sodium leaky channels. Phases and ionic basis of action potential in contractile tissues. So there are total 5 phases. So let's discuss all phases in detail. Phase 0 is also known as phase of depolarization. In this phase, our fast sodium channels open when threshold reaches to minus 60 to minus 70 millivolt, and this reaches to plus 20 millivolt. After that, our fast gated sodium channels are closed. Then the phase 1, which is initial repolarization, it begins because fast sodium channels are closed and fast gated potassium channels are open. Only repolarization begins in this phase. Then our calcium channels open, which occurs in phase 2, which is also known as plateau phase. In this, calcium ion permeability is increased, due to which our potassium ion permeabilities decrease. So combination of decreased potassium efflux and increased calcium efflux causes plateau phase. The calcium which is entering in this phase causing the activation of contractile process, but the actual contraction is caused because of the calcium which comes from sarcoplasmic reticulum. So these are phases 0, 1, 2, 3 and 4. In 0 phase, as we can see that potential from minus 90 millivolt reached up to plus 20 millivolt because of opening of fast sodium channels. Here fast sodium channels close and our potassium channels are open. When we reach this point, when in starting of phase 2, our calcium channels open but fast potassium channels are closed, so plateau is observed. Now phase 3 will start. Phase 3 is also known as phase of rapid repolarization because in this phase calcium channels are closed and slow potassium channels are open. Because of this, potassium ions rapidly exits the cell, and due to which plateau phase occurs. Now let's talk about phase 4. Phase 4 is also known as phase of resting membrane potential because now membrane has reached to its normal resting membrane potential. In this, because of rapid efflux of potassium ions, this causes to reach to resting membrane potential, which is minus 90 millivolt. Here we can see third and fourth phase. In third phase, our calcium channels are closed and potassium channels are open, so rapid repolarization is seen. And in phase 4, we can see flat slope, which is at minus 90 millivolt potential. So now next topic of slow action potential will be covered by Kamini. Thank you.
Hello everyone, my name is Kamini and my topic is action potential in conducting tissue. So first of all, what are the conducting tissue? Conducting tissue are basically the SA node and AV node. So slow response action potential is seen in SA node and AV node. What do you mean by slow response action potential? By slow response action potential, we mean that the depolarization is occurring very slowly. Now, why this depolarization is occurring slowly? There can be reasons like less number of gap junction, and there is a reason that the main channels that are responsible for the depolarization in the SA node and AV node is calcium channel, which are late to open and late to close. That's why the depolarization is slow, and it is called the slow response action potential. Now, the slow response action potential are characterized by a slow upstroke of influx of calcium and slower conduction than the fast potential, as we discussed above. Now, the resting membrane potential is about minus 55 to minus 60 millivolt. However, there is no true resting membrane potential because membrane never rests. Now, we will see this point in upcoming points. The slope of phase 4 of action potential is steep as compared to action potential in ventricular muscle fibers. जो फेज 4 होता है इसका, उसका जो स्लोप होती है वो फ्लैट नहीं होती है, वो स्टीप होती है. इसको हम आगे ग्राफ में देखेंगे. In SA node, there is slow but spontaneous depolarization occurring after every repolarization. This gives SA node and AV node the property of autorhythmicity. Now, this third point, which we discussed above, that the resting membrane potential is minus 55 to minus 60. However, it is not true. Why? In SA node, there is slow but spontaneous depolarization. It means that after every repolarization, there is slow but spontaneous depolarization occurring. So membrane never rests. Now the next are phases and ionic bases of action potential in SA node. There are total three phases: phase 0, phase 3, and phase 4. So firstly, we will talk about phase 0. There is high sodium ion concentration in extracellular fluid of nodal fiber. The positive sodium ion from outside tends to leak inside. यहाँ पे बेसिकली क्या बोला जा रहा है कि जो नोडल फाइबर्स होते हैं उनके जो एक्स्ट्रा सेलुलर फ्लूइड होता है उसमें जो सोडियम की कंसंट्रेशन है दैट इज वेरी हाई ड्यू टू विच द सोडियम टेंड्स टू एंटर इनटू द मेम्ब्रेन. The influx of sodium ion causes a slow rise in the resting membrane potential in positive direction. Therefore, the resting potential gradually rises and become less negative. When potential reaches a threshold of minus 40 millivolt, the L-type calcium channels opens, causing depolarization and action potential. We will see into the graph. So this is phase 0. Here थ्रेशोल्ड इज -40. जैसा कि मैंने आपको पीछे बोला कि जो एक्स्ट्रा सेलुलर फ्लूइड है नोडल टिश्यू के उसमें सोडियम आयन की कंसंट्रेशन इज हाई ड्यू टू विच सोडियम आयन जो है मेम्ब्रेन में आने लगता है जिसकी वजह से जो ये एक मिनट जो ये -60 पे यहाँ पे जो रेस्टिंग मेम्ब्रेन पोटेंशियल है ड्यू टू influx of the sodium ions, this reaches up to the -40, which is the threshold. Now at threshold the L-type calcium channels open, due to which action potential starts, and this is called phase 0. Next, now why the state of depolarization is not continued at all the time, like as L-type calcium channel could be open for a long time. So now we will answer this question. Now phase 3. Two events occur during action potential. Firstly, the L-type calcium channels becomes inactivated in about 100 to 150 milliseconds. So what is saying that the जो calcium channels होते हैं L-type channels वो open तो होते हैं लेकिन about 100 to 150 milliseconds के बाद वो close हो जाते हैं. And at the same time greatly increased number of potassium channel is opening. मतलब जो L type calcium channel है which was causing the depolarization that closes, and at the same time potassium channels that causes the repolarization that are opening. Therefore influx of calcium ion ceases and a large amount of positive potassium ion diffuses out of the membrane. Both of these events reduces intracellular potential back to its negative resting level and terminate the action potential. Now next, furthermore, potassium channels remains open for another few seconds causing hyperpolarization. पीछे हमने देखा कि जो पोटेशियम चैनल है दैट वर ओपनिंग विच वर कॉजिंग द रीपोलराइजेशन ऑफ द मेम्ब्रेन, बट दिस जो ये पोटेशियम चैनल्स है वो थोड़े फ्यू सेकंड लॉन्गर टाइम के लिए ये ओपन होते हैं जिसकी वजह से हाइपर पोलराइजेशन होती है मेम्ब्रेन की. अब जो ये हाइपर पोलराइजेशन है हो सकता था कि ये भी हमेशा के लिए मेंटेन हो जाए. अब जो पीछे हमने क्वेश्चन किया था कि जो डीपोलराइजेशन है वो क्यों नहीं हो रही थी मेंटेन हमेशा के लिए, वो उसके दो रीजन थे कि एक तो एल टाइप कैल्शियम चैनल्स जो है वो क्लोज हो जा रहे थे 100 to 100 मिली सेकेंड्स के बाद, और दूसरा था कि पोटेशियम चैनल्स ओपन हो रहे थे. ना जो ये हाइपर पोलराइजेशन है ये क्यों नहीं हो रही है मेंटेन फॉर एवर, इसका आंसर हम फेज 4 में देखेंगे. ड्यूरिंग नेक्स्ट फ्यू 10 सेकेंड प्रोग्रेसिवली more and more potassium channel closes. The potassium channel are closing here. Therefore, the hyperpolarization also causes inward flux of sodium ion that once again balances the outward flux of potassium and causes resting membrane potential to drift upward once again. जो पोटेशियम चैनल्स ओपन हो रहे थे वो फिर क्लोज होने स्टार्ट हो जाते हैं, ज़्यादा से ज़्यादा पोटेशियम आयन क्लोज होते जाते हैं जिसकी वजह से जो हाइपर पोलराइजेशन हुई होती थी वो क्या करते हैं कि जो इनवर्ड फ्लक्स होता है सोडियम आयन का उसको स्टिमुलेट कर देता है जिसकी वजह से सोडियम आयन जो है मेम्ब्रेन के अंदर आने स्टार्ट हो जाते हैं जिसकी वजह से जो रेस्टिंग मेम्ब्रेन पोटेंशियल है वो दोबारा से एक पॉजिटिव साइड की ओर जाने लगता है अगेन फाइनली रीचिंग द थ्रेशोल्ड. और जो अब अगर थ्रेशोल्ड पे रीच हो गया एक बार तो वो क्या करेगा एल टाइप कैल्शियम चैनल्स ओपन हो जाएंगे जिसकी वजह से दोबारा से डीपोलराइजेशन स्टार्ट हो जाएगी. नाउ हियर इज अ फैक्ट दैट these channels channels, sodium channels are called funny channels. This is because generally the sodium channels is open during the depolarization, as Isha has discussed before. But here, these channels are opening in a phase of hyperpolarization. Therefore, these channels are called funny channels, and the current is called as funny current, which contribute the earlier part of the pacemaker potential. And the later part of pacemaker potential before opening of L-type calcium channel is due to the opening of transient type calcium channels. Early part is due to the sodium channels, funny current, and the later part is due to the transient type of calcium channels. Now we will say it into the तो अब जो हमने अभी तक देखा है सारा स्लो रिस्पॉन्स एक्शन पोटेंशियल के बारे में, उसको हम ग्राफ में देखते हैं। सबसे पहले जो फेज 4 की बात करी थी हमने, फेज 4 में दो तरह के चैनल्स थे जो कंट्रीब्यूट कर रहे हैं, एक तो फनी चैनल्स और दूसरे ट्रांजिएंट टाइप ऑफ कैल्शियम चैनल्स। जो अर्ली पार्ट है फेज 4 का, क्या हो रहा था कि जैसे हाइपर पोलराइजेशन हुई तो उसकी वजह से जो सोडियम चैनल्स है दैट वर ओपन्ड, और जो सोडियम है वो अंदर आ रहा था फनी चैनल से। क्यों फनी चैनल्स क्यों बोलते हैं? क्योंकि हाइपर पोलराइजेशन के दौरान ओपन हो रहा है। तो जैसे ही सोडियम अंदर आया तो ये थोड़ा थोड़ा सोडियम उसको रेस्टिंग मेम्ब्रेन पोटेंशियल को हमारे पॉजिटिव साइड की ओर लेके जाने लगा। फिर लेटर पार्ट में ट्रांजिएंट टाइप ऑफ कैल्शियम चैनल्स ओपन हो गए जो कि हमारे रेस्टिंग मेम्ब्रेन पोटेंशियल को -60 से -40 तक लेके जा रहा है। अब अगर एक बार थ्रेशोल्ड रीच हो गया तो यहाँ पे लॉन्ग लास्टिंग कैल्शियम चैनल्स ओपन हो जाते हैं, जिसकी वजह से डीपोलराइजेशन होगी और एक्शन पोटेंशियल भी आएगा। अब जो ये एल टाइप कैल्शियम चैनल थे ये कब बंद हो रहे थे? 100 टू 150 मिली सेकेंड के बाद ये बंद हो जा रहे थे, जहाँ पे कि फिर पोटेशियम चैनल्स ओपन हो रहे थे। अब अगर पोटेशियम चैनल्स ओपन हो रहे हैं और कैल्शियम चैनल्स बंद हो रहे हैं, मतलब कि पोटेशियम का आउटफ्लक्स ज़्यादा होएगा और जो कैल्शियम चैनल्स बंद हो गए हैं तो मतलब कैल्शियम का इनफ्लक्स स्टॉप हो जाएगा। अब पोटेशियम का आउटफ्लक्स हो रहा है, आउटफ्लक्स होता जा रहा है, तो पॉजिटिव जो आयन है वो मेम्ब्रेन से लॉस हो रहा है, जिसकी वजह से हमारे जो मेम्ब्रेन पोटेंशियल है वो दोबारा नेगेटिव की ओर जा रहा है क्योंकि पॉजिटिव लॉस हो रहा है। अब ये नेगेटिव की ओर जाता है, लेकिन जो हमने पढ़ा था फिर फेज 3 में कि जो पोटेशियम चैनल्स है वो थोड़ी ज़्यादा देर तक ओपन हो जाते हैं, जिसकी वजह से हाइपर पोलराइजेशन हो जाती है। फिर यहाँ से दोबारा से वही फेज कंटिन्यू हो जाएगा। हाइपर पोलराइजेशन होगी तो सोडियम चैनल ओपन हो जाएंगे, सोडियम चैनल फिर से ओपन हो गए, देन बाद में ट्रांजिएंट टाइप कैल्शियम चैनल्स ओपन होंगे, फिर से ये नेगेटिव -60 से रेस्टिंग मेम्ब्रेन पोटेंशियल को थ्रेशोल्ड पे लेके जाएंगे। फिर वहां से दोबारा फेज 0 स्टार्ट होगा, डीपोलराइजेशन होगी, फिर रिपोलराइजेशन होगी, फिर हाइपर पोलराइजेशन होगी। तो ये जो साइकिल है ये एक जो भी इंसान होता है उसकी लाइफ टाइम चलती रहती है, एंड दिस इज हाउ द स्लो एक्शन पोटेंशियल। थैंक यू।